to introduce our speakers. I'll introduce both at the beginning so as not to interrupt the flow as they go from speaker to speaker. So the first speaker today will be Dr. Hannah El-Samad, and she is a professor of biochemistry and biophysics at the University of California, San Francisco School of Medicine. Her focus is on the basic design principles of biologic signaling, and that includes inter and intracellular communication and processing of single and multicellular systems, evolutionary rewiring of conserved pathways across multiple species, and population heterogeneity in cellular decision making. Our second speaker today will be Dr. Oscar Sess, and he is a professor and the head of the Department of Chemistry and professor of chemical biology at Imperial College London. His interests in the area of synthetic biology are the construction of artificial cells using microfluidic technologies or bottom-up synthetic biology and molecular membrane engineering. And so we've got a terrific webinar for you, and I hope you enjoy it. And our first speaker will be Hannah.
Thank you, Stephen. And good afternoon, everybody. Thank you all for being here. I know that you're all busy homeschooling your kids and making sure that yourself and your labs are well equipped to deal with the repercussions of this pandemic. So I really appreciate your time. And thank you to SCBM for organizing this series and for this invitation. So for those who know me, I am an engineer by training, and I have been fascinated for a few decades now by how biological circuits work. So I sincerely and wholeheartedly think and believe that underlying the existence and persistence of everything that is living is the use of feedback. And feedback, as you know, is the ability of a system to sense either its own state or the environment, and to compute and correct accordingly. And to do that, of course, not only once, but over and over again in real time for the span of its life, or, for technological systems, of course, this is the use of feedback is a very common occurrence. This is why we can fly planes and so on. So feedback of this kind is present, of course, at every level of biological organization. We see it inside cells, among cells, and all the way to the physiological level, and of course to the ecological level. What we're experiencing in California right now is unfortunately a starting to see, in terms of fires and climate change, is starting to see that homeostasis or equilibrium established by feedback start to loosen up a little bit. And of course, what I'm saying here is not new to any biologist or student of nature. So students of nature for a long time have understood that feedback is really important for homeostasis. On this slide, I have the book that really influenced my thinking when I started, transitioning from engineering into biology. The Wisdom of the Body by Walter Cannon, who was a medical doctor. So while my lab does quite a bit of things, ranging from systems to synthetic biology, at the heart of it, what we keep trying to answer is the question of what do feedback loops in cells and in general in life do? And I would like to make that provocative statement now that while cells, even the simplest of cells, have thousands of feedback loops, the ones that we really understand are not very many. So in my lab, we find ourselves asking whether we can understand feedback loops, whether we can understand their failure modes, trying to fix them, whether we can build them de novo with synthetic biology techniques, and also whether we can devise new and improved mathematical frameworks that capture their fundamental principles. I will touch upon these, but not some of these, but not all of them today. So to start, I would like to go back to the ancient times, and that's 2011, when we and our collaborators at ETH Zurich asked a very simple question, which is, can we take one gene inside one cell? In this case, it was the yeast Saccharomyces cerevisiae, and exert external control over it. And what I mean by that is that can we take that gene and drive it through external feedback control to a place where it doesn't want to go, to a level of expression that is not native to that gene, and keep it there through feedback control, despite the cell trying to fight back its way to return to its physiological level. And of course, candidly, there was no really deep scientific question that we wanted to answer that. It was just, a game we played, and the question was really, can we even do that? And this is actually a theme in my work. We try things just to kind of play daredevils sometimes. So what we did to do that is the following. We built an automation setup in which we measure the expression of that gene through a fluorescent reporter, then feed the readout into a computer that is running an algorithm. So it compares that measurement to the desired value that this gene doesn't want to assume, but we want it to assume that. So it's the desired value. It computes an error signal. So it's a difference between where the gene expression is and where we want it to be. And then feeds it through a control algorithm, then determines how much of a stimulus the cell needs to be given in order to induce sufficient gene expression to regulate to the level we want. And then this command is kind of passed to the cell. So how this command is passed to the cell is through the use of optogenetics. So we engineer that gene to be light responsive. And then by pulsing the cell with light, we can, so to speak, remote control it. This is, of course, this whole process I just described to you is, of course, repeated every few minutes. And every time that gene is trying to escape back to where it wants to be, through that process, we can push it back to our desired level through zapping the cell with light based on the output of the control algorithm. So this is published. I will not go into to a lot of data. I will just show you one slide of data, which is, this is the gene expression of, actually it was the GAL gene, one of the GAL genes. And these are 4 cultures that we wanted to regulate to some arbitrary level in this slide. It's a level 5 of arbitrary, fluorescence units. And as you can see, these 4 cultures want to be at a different place, and through zapping with light and doing this feedback control, we can actually bring it to 5 and keep it there forever and ever. So that was fun. That was great. It was like playing video games with cells. But also inside that video game was a quite a serious actually question. And the question was, can we use the same strategy, that external feedback control of cells, to fix a cell that has its own broken feedback in the same way? So use the same technology to fix a broken cell. So we to test this question, we turned to a cellular pathway that has many feedback loops. And this is the mating response, or the pheromone response, in also the yeast Saccharomyces cerevisiae. So this system has many, many feedback loops. It's a MAP kinase pathway that responds, as I said, to pheromone. I will really not discuss the biology of the system, and just try to give you the rationale of our study and the gist of our finding. So here's what we did. We took the intact system and gave it an input of pheromone, or alpha factor, and then looked at its output, in this case, a gene promoter measured using a fluorescent protein fusion as a function of time. Then we deleted one of its feedbacks, and in this case, it's feedback through the phosphatase MSG5. And we repeated the same experiment. We gave the input and looked at the output. so this is how, first of all, this is how the data looks like. So this is the output plotted as a function of time for a dose of pheromone. And as you can see that the wild type and the mutant are obviously different. Now, what's important about this is that the difference between the mutant and the wild type is actually not constant. It's changing as a function of time. So if we were to ask, what would it take to correct that pathology? How much should we put back the mutant gene? How much does the mutant gene need to express in order to compensate? How much do we need to express MSG5 inside that cell to compensate for the lack of feedback? The answer to that is that you cannot just put some amount, constant amount of MSG5. Correcting that difference between the wild type and the mutant has to be dynamically programmed. So we need to put in different amount of MSG5 back as a function of time in order to make the mutant now look like the wild type. So that looks like a really good application of this optogenetic control setup that I showed you before. And this is exactly what we did. We asked, can we put this cell that has this mutant and wild type pathways, two cultures inside that setup, measure the wild type and measure the mutant and say, can we actually correct the mutant through a feedback control in order to bring that trajectory of the mutant to that of the wild type? And of course, we could do that. This is why I am telling you this story. So we could use that in silico feedback control to correct the pathology. So this is great, but the real question, the real interesting thing here is what was the program? What was the optogenetic input as a function of time that was needed for this correction? So in this case, it was actually a set of pulses separated by 1 hour. So of course, there are many biological implications for this result, for this particular gene and so on. But in general, we were very excited by this as a way to start thinking about a method to read out feedback dynamics in real time. Because a process such as feedback that is inherently dynamic cannot be understood by taking a static snapshot. And in this case, this synthetic scheme involving control by a computer can be used to start, as in this case, showing you what is the time scale, what could be the time scale and a contribution of a feedback loop such as that of MSG5. So I'm, of course, here I'm showing you only one experiment, only one example, but one can generalize this and figure out a bunch of neat things, and of course we did in that paper, whose reference is given at the bottom of the slide. So I'm just giving you really a cursory view of how a kind of a simple-minded quest to control a gene through feedback control ended up giving us a technology that hopefully will allow us to study feedback. Or at least generate hypotheses about feedback control in cells at scale. So along this trajectory, and when we were programming computers to control cells and so on, we started thinking that the next step should actually be the ability to build feedback control inside cell from the ground up with biological molecules. And of course, serendipity is just an amazing force in science, because just around the time when we were thinking about that, I visited the University of Washington, Seattle, where I met with David Baker. Of course, you heard through this seminar series also from David a few weeks ago, I believe. So for those who don't know David, him and his crew are really pioneers in understanding how protein fold and also creating protein forms that fold and work from first design principles, generating constructs and proteins that are not variations of what exists in nature, but truly de novo forms. So for a number of years now, we have been working with them to to see whether some of their de novo proteins, some of the classes of the designer proteins they produce could be actually used for synthetic biology. So in my mind, I mean, of course, there's many brands of successful synthetic biology and cell engineering out there. And it is something that we also dabbled in. But in my mind, the use of actually de novo designer proteins for synthetic biology has many advantages. They're simpler than evolved proteins. They potentially have much less crosstalk with the cell. And they are unifunctional. They're designed to do one thing and do it really well. Of course, if the design is successful, in contrast to evolved proteins, which are multifunctional, complicating, of course, their use for synthetic biology. So one such designer protein is, and here's David and Bobby and Scott, who from his lab, who were our collaborators in this, and from my lab, Andrew was really the pioneer in the next few slides, the story of the next few slides. So one such design is a protein called LOCKR, which is a de novo designed bundle of 5 helices held together through a designed hydrogen bond network. So I will call this a switch. And a 6th helix, which I will call a latch, is designed to connect to the other helices through a hinge. And this latch is destabilized by design by mutating certain of its residues so that it can be dislodged from the rest. So it can open and close. That latch can open and close. And a single peptide, which is shown here, can then be threaded into that latch helix so that it's hidden when the latch is closed and exposed when this latch is open. And this is the depiction of that. So there's a latch that closes and open, and the bioactive peptide, and I'm going to tell you in a minute what this bioactive peptide we're using here is exposed when the latch is open. Now, this latch is open when a genetically encoded key molecule, which is also designed, is presented to the switch. So I will really not dwell on details other than that, because this work is also published. So from now on, what I will do is that I will designate the switch as this blue thing here, the key as this pink circle, the latch is this thing I'm pointing here. Hopefully you can see my cursor. And then the peptide molecule is, the bioactive peptide is this red thing here. So when it opens, that bioactive peptide is exposed, and then you get functionality. Of course, this bioactive peptide can be really anything. I will focus on only one functionality, which is a degron peptide, which is when revealed by opening of the switch, leads to the degradation of that switch and any cargo really appended to it, any other molecule appended to it. This is why we call it the degron locker. And here's some data showing that as you present more and more key to the switch, it disappears. So it gets degraded through a fluorescence assay. Now, this cargo that you can stitch onto the degron locker molecule or the switch could be really anything. It could be a transcription factor, it could be an enzyme, it could be a kinase, it could be any biological molecule. And as I said, as this switch is activated, the switch and its appended molecule are degraded. I mean, of course, so that leads to regulation. You can imagine how that can lead to regulation of signaling in pathways. Now, of course, this can be achieved for any degradation systems that are triggered, for example, with small molecules, and there are many beautiful examples out there of this. But if the aim here is to build synthetic circuits, then one has to make the trigger, of course, genetically encodable, that is produced by the cell. And this is actually my favorite thing about the degron locker system. The fact that the peptide, the key, is a peptide that is also produced by the cell means that it's networkable into circuits. So of course, the first thing, this is why I'm telling you about this, the first thing I wanted to build with this is a feedback circuit. And it actually doesn't take much imagination to figure out how. Let's say we have a pathway, A, B, C, where we want to induce feedback on the node B. So we just stitch B to the degron locker switch. And then to induce feedback, we go now and produce the key from the output of this pathway, let's say node C, and you've got yourself a negative feedback loop this way. So, of course, we went ahead and built this. And the first, I'm just going to give you really like one set of data on this. And of course, this work is also published. So if you're interested, you can go ahead and delve into the details of the publication. So we really, as an engineer, I really wanted to benchmark this. I wanted to first test it in a synthetic context where I can understand its limitations, what it can and cannot do in a really engineering sense. So the way we vetted it is it's truly in a synthetic context. So building a feedback loop, like literally in a two-node circuit, which is shown here. It's the notation here is on the right here is the actual circuit, and it sounds really complicated, but what it is what's shown on the left, which is two transcription factors A and B that are cascaded in series. Now, the experiment to test the feedback here would be if I basically generate, if I give the circuit an input, generate an output, and the system settles at this output, and I came and shake that circuit. So I added disturbance. What happens to the system with feedback or without feedback, right? So this is testing the classical notion of homeostasis through feedback. And this is precisely what, I'm going to show you the data for this. And this is precisely what we did. This is the system first without feedback. So we give an input. For that input, the output settles at some level. Once it's reaches steady state, we go and disturb through a small molecule the node B, which is here transcription factor that is actually receptive to that molecule. So it's a hormone inducible transcription factor. And of course, a system with no feedback is completely helpless to its environment. So as you disturb it, it goes and settles at a very high, a different steady state than where you want it to be. And then, of course, this is the classical disturbance from homeostasis for a system that has no feedback. Now, let's add the feedback. So let's put this, let's append the Degron switch to node A. Let's produce the key from the second transcription factor B, and let's repeat the same experiment. And this is, of course, what the data look like. Of course, you can see that the system, when it's disturbed, it has to respond. Of course, it's been affected, but the question is, does it recover? And in this case, the feedback helps it recover. So this is what feedback is really about, right? It's helping make things, circuits from technological to biological circuits robust to the world around them. So of course, that same feedback strategy that works for a synthetic system works for a complicated endogenous system. And I'm not going to dwell again on this slide. It shows that if we go back to our friend, the mating pathway in yeast that I showed you in the beginning of this talk, and now we induce a synthetic feedback in it. So now we're not disturbing it. We're not killing any of its feedback. We just add another feedback layer on top of this. We can actually induce very different behaviors in the output. So we can make it now adapt instead of just go to a to just a steady state it wants to go to with the no feedback, meaning its own endogenous feedbacks. But we can also, because this is all these molecules are designed molecules, so we can change their affinity to each other. We can change the design, and we can actually modulate the impact and qualitatively and quantitatively of the feedback that we can induce on this pathway through that. And this is another kind of advantage of designer molecules, that you can actually modulate these properties in a semi-rational way. So of course, feedback, and I'm almost done here. I'm just going to give you just my final thought. Of course, feedback is not the only thing we can build with these designer molecule, and we're in the process of building a whole signal processing toolbox with these molecules, and it's been such a fun ride. It's just been amazingly fun. So the question is, the final question here that I'm going to address is, so what? So what can you do with that? Why is that even useful? I'll just give you one possibility. So even though we actually dabbled in synthetic biology and cell engineering for upward of 15 years now, I actually started being only serious about it a few years ago, after the notion of live cell therapies became a reality. So this is an FDA-approved T cell that was approved in 2017 for B cell to treat B cell lymphomas. And what this is actually a really, I mean, it was just an amazing moment, actually, I think, for medicine. So this is called adoptive immunotherapy, and it's the concept of which is that you can take cells out, T cells in this case, out of a body of a patient. You can change them. You can engineer them so that they have, in this case, chimeric antigen receptors, or CARs, to recognize an antigen on a B cell. And this recognition, when you put these cells back into a patient, leads to activation of these engineered T cells to do what these T cells do, which is kill the cancer cells. So this was a truly breakthrough moment. But it was, for those of us in engineering, it was a moment that resembled when the Wright brothers showcased their first gliders. It was a monumentous, event. We learned that we could fly. I mean, this is amazing. We could fly, but we also know that we can crash. So these engineered cells, of course, remain very dangerous. They're quite primitive. They're reserved as such as weapons of last resort. These are not your everyday drugs. These are the drugs that are used when everything else fails. So the question is, how can we change that? How can we make them safe, smart, robust, reliable enough in order to deploy these engineered cells to really harness the power of programmable biology? Again, not as a measure of last resort, but as something to treat chronic conditions, for wound healing, for resetting circadian rhythms, for those of us who used to travel and had jet lag, right? It is really interesting to think about living cell therapeutics as as the future. So what needs to be done, at least to me, is really to make that kind of pipe dream a reality, is of course rational designs of circuits that work well, that have feedback control so that they're not helpless to the environment of the body. We also need theoretical understanding of biological molecule as new engineering substrates. We're dealing here with biological molecules, with biochemistry. We're not dealing with electronics. Simply said, I think the rational design of synthetic circuits has finally found its calling. So we really hope that the feedback circuits we're building and so on will move the needle a little bit on that. It's going to be a long journey, but it is exciting. We can fly. Remember, now we have to build the jetliner. So with that, I will stop and thank the people who actually did the work for the 2011 paper on playing video games with cells. This was done in collaboration with John Ligeros and Mustafa Kamash at ETH Zurich, and spearheaded by Jacob, David, and Ignacio. The champion of the closed loop feedback control to correct cellular pathology is Patrick, and this work was done with Hitan Madani at UCSF. And of course, I mentioned that the de novo design of proteins and the synthetic biology done with that is done in collaboration with David, Scott, and Bobby at UW. And the stars of this are Andrew, Galen, and Taylor. And thank you very much. I hope I didn't go too much over my allotted time. These are the people as an entity who funded us. So thanks of course to them for humoring our playing games. And with that, I will cede the floor to Oscar.
Thank you very much, Hannah. Oscar, you're up.
Well, I'm just going to share my screen a second. Hang on a second. Steve, can you see my slides?
Yes, yes, I can.
Okay, let me start in the right place. Brilliant. So, afternoon, folks. It's a real pleasure to be here today. Really appreciate the invitation. Also, just want to say, make sure everyone's kind of keeping safe. So, uh, just really enjoyed the previous talk, and I'm going to follow in a kind of similar kind of spirit, but with a, I'd say, even more bottom-up, minimalistic kind of approach. So, just a little bit about my group. So, we're based at Imperial College in London. So, in the top corner, you kind of see where the main university is based in central London near the museums, but we actually moved a couple of months ago, about 2 miles due west to a new campus at White City. The chemistry department moved there, and I'm lucky. I kind of run a large group with my collaborators. You can see the photo there: Rob Law, Nick Brooks, John Senyval, and the kind of group together looks at 5 kind of areas. So synthetic cells, optical nanofluidics, microfluidic technologies for studying the protein content in single cells. We do a lot of work around membrane protein interactions, which will feed into today's discussion. And also, we look at how lipid assemblies kind of function. But today's talk will bring together work on synthetic cells and membrane protein interactions. So, from my point of view, so I'm a chemist by training. When I think of the area kind of synthetic biology, you've just seen just now how astonishingly powerful it is. But with my chemist hat on, I always make an analogy between a cell and a city. And if I use the analogy with London, London has a big motorway around it called the M25. And, I often think of kind of roads in the city as kind of molecular pathways, the boroughs and are equivalent to kind of organelles, and then the cars and the people, they're equivalent to kind of molecular traffic. And I often say to my students, imagine an experiment where I said, look, let's try and control London. And you kind of go, right, let's block the traffic going from the main motorway, so for the main airport, Heathrow Airport, to the centre of London, Piccadilly Circus, and we'll get in a car, we'll drive, we'll get out of the car, we'll sit on the motorway, we'll lie down the road, and we'll block the traffic, and London will respond. Traffic will build up, but eventually, most people get a bit annoyed, and they'll wonder why you're doing that anyway. But they'll get their phones out, you look at Waze, they'll look at Google Maps, Apple Maps, whatever you use, and they'll eventually circumvent the traffic. So at that point, a macro object, which you can see, is already quite hard to control, and that's a situation where you've got 8 million different moving parts. You take a cell, imagine if you then miniaturize London and you made it 10 microns in size, and rather than having 8 million different objects, you've actually got about 3.5 million different types, let alone total copy number. You can see how complex the challenge can be. And just now you've seen some really beautiful work from Hannah about how you can make a cell behave correctly, but at the same time, it's still a very complex system. So we thought, actually, can you make new London? Can you make one from first principles where actually in this version of London, you only have the outer boundaries, the motorway, you build the airport, you build the centre of town, Piccadilly Circus, and you build one motorway between those two. And in that version of London, which is very simplified, let's call it New London, when you block that motorway, London behaves perfectly because it cannot do anything else. You put in the one circuit, the 2 circuits it needs, and then you kind of build up from there. So you basically put in very simple parts using very simple components. And what we try and do in the group then is engineer behaviors building upon that. So it's kind of a very minimalistic bottom-up synthetic biology kind of approach. So basically what we do is develop technology in the group to basically make the outer chassis of synthetic cells and then actually introduce compartmentalization. We try and reconstitute different types of proteins, either peripheral or integral, and make sure they kind of have full functionality. Then we try and introduce different subcompartments into these synthetic cells. And then we try and get these components to kind of work together in tandem to produce basic behaviors. And I'm talking really kind of basic, movement, information storage, chemical kind of synthesis, environmental kind of sensing. And then as we engineer those different behaviors, we try and stitch them together to get increasingly kind of more complex kind of systems. But the first thing we had to do as a group was basically engineer the technology to actually kind of make these compartments, dial up a synthetic cell of a given size, of a given level of compartmentalization with a given level of composition. So where we've got to now as a field, not just our group, is that these are kind of architectures you can make now. You've got the traditional kind of vesicle, but now you can make multi-compartment vesicles that are phase separated in terms of subvolumes, large-scale tissues made of droplet interface bilayers. I'll talk about later on a bit more. Multisomes. These ones here, vesicles within vesicles where they're concentrically predisposed, or nested kind of vesicles where you've got smaller ones inside larger ones. All this kind of architecture, I'd say 10 years ago, were kind of really kind of beyond the realm of what most people could make on demand. But now we've got to the point we can use these. You can then start to put in circuits inside more proteins and start to engineer functionality. The other thing I think that's changed in the field in recent years is most of the things that we make, we use using microfluidics. so kind of, you know, lab on a chip type technologies. And again, say 10 or 15 years ago, you would need a clean room worth a couple of $1 million to kind of enter the field. But it's now got to the point where on a benchtop for about $1,000, you can be really quite active in the area of microfluidics. So we spend a lot of time out of the clean room trying to make sure that any approach we develop, you can replicate on a desktop for a couple $100 to make sure that kind of microfluidic approach is widely available. So on the one hand, for example, here, we use a desktop laminator to put down multiple layers of dry film resist. Then we use, you know, UV polymerization to make the channels, and then we kind of seal it. That approach is talking about $1,000. In other cases, we basically kind of use laser cutters to actually etch the channels. But in all cases, you don't need a really expensive clean room to actually start to make droplet-based objects on the length scale of these kind of channels here are roughly anything between 10 to 100 microns. And as I said, you know, it does not require a clean room to do that. So here's an example now of the kind of way we use these approaches. So here's an example of a microfluidic chip, and the basic architecture I showed you at the beginning was, you know, you have to make the outer chassis for this kind of bottom-up synthetic cell. So what we've got here on the left-hand side is you've got an aqueous junction in the middle, and you've got two oil-based junctions coming from top and bottom. So obviously, when you bring those co-flows together, it basically produces a series of water droplets in oil. This is normally at kind of kilohertz kind of rates. But what's special here is the oil has lipid dissolved in it. So the lipid then basically goes to the interface of the droplets to produce a monolayer. By the time it then enters the serpentine, those droplets that are meandering through the serpentine, the monolayer interface is stabilizing around them. And then at that point here, when they exit the chip, there's a little step there that makes the droplets now hop from an oil volume into an aqueous volume. And the aqueous volume also has another lipid type, and that lipid type marries onto the monolayer to produce a bilayer. So if the two lipids, the lipids in oil and the water solution, are identical, you get a symmetric bilayer. If they're different, you get an asymmetric bilayer. So you can slightly dial up, and also depending on the flow rates you use and channel geometries, you can change the size of the vesicles, but you can also change whether you reconstitute proteins in real time, and also whether the membrane is symmetric or not. So there's a lot of functionality, and we use a variety of techniques this way, also microfluidic kind of focusing and so on. Now, what we then did was, as I mentioned, the other part of the group looks a lot at membrane-protein interactions. And we were thinking, you know, can we start to make very, very, very basic biological circuits based upon the following principle, which is, if you look in the literature, there are literally thousands and thousands of papers which look at how membrane properties regulate protein function and vice versa. So, you know, for example, how rigid a membrane is, how curved a membrane is the membrane asymmetric, and so on, you know, its fluidity. And then people discuss how that influences the function of proteins that are either embedded or in contact with that membrane, and vice versa. And there's some, you know, really, really beautiful work in this area. And we kind of, you know, and that was an area that we kind of focused on for many years and still do. We kind of took a step back and we said, hang on, if a protein can sense a membrane, a bit like, you know, the days when I was younger before mobile phones, you know, you kind of get two paper cups and a string, you know, you'd shout down one paper cup, and the other person with the other paper cup, the other end of the string could hear you. You know, the signal was transmitted by the string. Could you use a membrane to propagate a signal between two proteins that are not in contact? So when you often think about protein-protein communication, you know, people think about two proteins docking together and there being a kind of a follow-on process. But actually we thought, hang on, if protein one comes down to the membrane, it changes the composition, all the mechanical properties of that membrane. Can another protein that is not in contact with that protein sense that and respond accordingly? And if you can do that, can you then start to use that to engineer functionality and behaviors? So on this screen here, you get some examples of bacteriodopsin, a protein pump. Here's a mechanosensitive channel, and this one's basically like a channel protein that sits in a membrane, and it opens and it closes in response to asymmetry. And then there's phospholipase here, which is a protein that when it sees a membrane, it dives into it, it skirts along the surface and catalytically chops off the tails. So we thought, can we get, in the first one, I'll show you now in a movie, could we get phospholipase to talk to MSCL and use that to make a synthetic cell that opens up in response to an external stimuli? So I'm just going to play a movie now. Hopefully you'll see it there. So if I just pause that a second, in this case here, this machine you'll see there only has about 6 or 7 molecules. So the vesicles themselves have about 3 or 4 lipid types that are required to make the mechanosensitive channel functional, and all the membranes are initially made asymmetric. So as you zoom in on the surface, you kind of see the lipid leaflet, And here you'll see the mechanosensitive channel, MSCL, embedded in the membrane, and it's initially closed because the membrane it sits inside is asymmetric. And then when you see now the cargo, it can't escape because basically the channel's closed. But now if you add phospholipase, the other protein from the outside, it can only attack from one side. So basically it jacks into the membrane, it picks up the head, it starts to chop off tails, and because it can only do that from one side, if the flip-flop rate is low enough, you've made the membrane asymmetric. That asymmetry propagates in a very simplistic kind of way. MSCL senses that, and it opens up and it releases a cargo. So you've suddenly got this very basic machine that it will stay closed unless phospholipase binds to the membrane. And it's really chemically specific. If you change the mechanics of the membrane, the rate at which the two proteins talk to each other can be modulated. And also, if you change the lipid type, so the phospholipase needs an ester group to actually carve the tail off. If you change it into an ether link, nothing happens. So you can basically have both analog control and digital control of the rate of communication between the two proteins. And then we use that approach to actually then start to build up a synthetic signaling cascade. So here in the top left, we make a nested vesicle system. So in the inside vesicle, you've got these nested vesicles, and basically they have MSCL reconstituted within them. And then in the lumen of the vesicle, it turns out that phospholipase that you saw that was attacking the membrane, it needs calcium to be active. So the inside lumen of the larger GUV is swamped with EDTA. So EDTA takes up all the calcium that's needed. So you've basically got the phospholipase that's dancing around inside the lumen. It's attaching itself to the membrane, but it won't actually make the membrane asymmetric because there's no calcium there. And in the meantime, the MSCL remains closed. But then what you then do is if you add alpha hemolysin, which is basically a channel protein that when it sees a membrane, it basically dives into the membrane and it forms a pore. What happens then if you've got an outside barrel of calcium, the calcium goes on the inside, it swamps the amount of EDTA that's on the inside, the phospholipase becomes active, the phospholipase attacks the inside membranes, they become asymmetric, the channels open, and you release a cargo. And that worked really beautifully, and the amount of calcium that you pump into the cells is then proportional to how fast the signal propagates. And these synthetic kind of, the inner vesicles, they've basically had a fluorescence kind of system that basically leached outwards, but the rate at which you saw that happen was directly proportional to how strong the signaling cascade was. And again, this system, you're talking 7-8 different types of molecules, so very, very simple systems, but we know how each molecular component works, so it's relatively easy to stitch things together and get kind of predictable kind of behaviors. Then what we've done down the line is, in terms of a slightly more controlled kind of way of deciding how one of these synthetic cells, how fast it leaks, is again, this is done on chip. So you think of it as a production line where as the vesicles are coming off chip, they've got these alpha hemolysin pores inside them, and inside them, but using microfluidics, you regulate the concentration of trimeb. So trimeb is like a blocker, which when it sees the pore, it blocks it and it hops on and off. And by basically in real time changing the concentration of the blockers, but keeping the number of pores constant, you can fine tune how leaky or unleaky you want a vesicle to be in real time, which again was really nice in terms of trying to engineer smart delivery systems. Now, at the start of the talk, I mentioned wanting to introduce compartmentalization. So the way we tried to engineer that is building upon some really lovely work years ago by Hagen, Bailey, and Oxford, and also Shogi Takeuchi in Japan. And they basically kind of put together a system which, commonly called, droplet interface bilayers now. And the way I think of it is kind of like lipid Lego. So what you have is a series of droplets in oil. Again, you have lipid dissolved in the oil. You form one layer to drop the free energy. And at the point of contact, when you plug these two together, you form a bilayer. So here you see a series of, you've got monolayer, bilayer, monolayer, bilayer, monolayer, bilayer in oil. And if you make these in a stable kind of way, these can sit on a windowsill for weeks and months. They're really, really robust kind of systems. But the point is, when you plug them together, if the lipid monolayers are the same, you form a symmetric bilayer. If they're different, you form an asymmetric bilayer. And here on the bottom side, you can see kind of a droplet bilayer network. Here we have five different droplets, and you've got 4 different bilayers connecting the central one to the outer droplets. And again, in this case here, this was just made with a 3D printer. So you're talking £200 or £300 auntie maker, and you're in business with making droplet interface bilayers. In terms of using that to make kind of larger systems, we use, again, microfluidics and robots. So we actually have a robot that's 32 different wells, and you can aspirate different compositions of lipid to make different kind of droplets. and then you can stitch these together to make kind of large kind of architectures. This is kind of what we're trying to do here is replicate the kind of membrane translocation pathways that you'd see in real kind of tissues. But in all these cases, actually, I should go to the next slide first. As well as using kind of fluid to move things around, we also kind of use optical traps. So optical traps are basically like, basically using a focused kind of laser beam to move objects around. So in that case here, rather kind of using in the previous slide fluid motivation to make kind of networks of droplets that form membranes, here we kind of use light to make, here it says IC, O for Oscar, linear kind of networks, 3D kind of networks. But this is a low throughput way of doing it. But the principle was just to kind of show that we can make kind of really nice kind of architectures. A slightly kind of more quirky application, which I just thought I'd show you for fun, was in this case here it was surfactant based rather than lipid based, was to make basically a bio TV. So what you've got here is a series of kind of droplets, which again are connected, and in each droplet you have a cage dye, and when you shine a confocal pulse on an individual droplet, the droplet acts as a pixel, and you basically release the dye locally, and you can basically draw an image or erase it and kind of reproduce systems. So it's basically, and this system here, you're talking 4 or 5 molecules to make the world's most basic biological kind of television. Now, back to compartmentalization. If I just go back here, all these systems are lovely, but the water droplets, which have these monolayer, bilayer coatings, are ultimately in oil. If you want to make a synthetic cell, you have to have an outer bilayer. So what we then developed was approaches for both on-chip and off-chip for taking these droplet interface bilayers out of the bulk oil, passing them again through an oil-water interface. And when they go through an oil-water interface, that adds a monolayer onto the already present monolayer, so suddenly then produce a compartmentalized vesicle. So if you look at this kind of system here you have a 2-compartment synthetic cell. You've got protein expression machinery for red and green fluorescent proteins in each compartment, and that's expressed differentially. The 2 compartments can't talk to each other, but previously that was made by having, as I said, 2 droplet interface bilayer in bulk oil. You pass that through an oil-water interface where you have lipid, it coats it, and you form a bilayer. And we've been able to, the highest we've got up to is about 20 different subcompartments, which you'll see later on, but we can make synthetic cells with 2 compartments, with 3 compartments, with 4 compartments. We can control the composition, both lipid-wise and protein-wise, at each interface and each subcompartment. So the next thing we did was, in terms of engineering kind of user-defined cascades, we said, right, if we can make these systems now, can we decide what goes in the lumen of each subcompartment and engineer user-defined signaling cascades? So we did that, and we kind of made a series of compartmentalized systems where in this case here, you had beta-glucosidase in one compartment, glucose oxidase in the next, phosphatase peroxidase in the last compartment. And then I showed you before alpha hemolysin. We basically constructed these where in some cases we had alpha hemolysin as a set of pores between different kind of interfaces to allow the reactants to flow in a certain direction, or you change the order of the enzymes, and only when you had the right order of the enzymes and all the compartments connected can you have a situation where the lactase comes in and eventually is basically processed by the enzymes to produce ricirufin. But the key thing here is the enzyme cascade, and the chemical you produce is defined by the order and the sequence of the different enzymes you can put into these different kind of subcompartments. So that was a really nice kind of piece of work we did a couple of years ago. Now, I mentioned at the start that one of the things we wanted to do was, mimic compartmentalization, but also the kind of architectures that you kind of see in nature. And an architecture which you regularly see in nature is that of the immune synapse. So here in the bottom left image, you see what appears to be a droplet interface bilayer. So it looked before like it was a monolayer. Here's a bilayer. Here's a monolayer. Actually, what this is are 2 vesicles that have come together and they've snapped together. So if you look at the video on the right-hand side, you'll see in a second, an optical trap will pick up one of the vesicles. And if you control basically the ionic strength in solution, you can regulate the interaction with the 2 vesicles. In this case, if you get it just right, when you bring the 2 vesicles together, they snap and they form a double bilayer. So that is they've not fused together to form a single membrane. That is 2 membranes that are literally nanometers apart, just like you'd seen it in an immune kind of synapse. And then again, using the laser beams, you can then start to make kind of more complex kind of architecture. So here you see the optical trap picks up a second kind of vesicle to form another VIB, picks up a third one to form a second VIB, and then a fourth one. And now if you rearrange these, you can basically make kind of a tetragonal kind of structure. And these are really stable. While you basically keep the ionic strength fixed, you can't pull the VIBs together. They literally roll upon each other like you would like a zip. But then if you change the ionic strength, they'll actually kind of fall apart. So here in the video, you basically see them being used to form the pyramid structure in the top right corner. You can then use that to also do chemical synthesis in a control kind of manner. So when we started making these, we then eventually introduced biotinylated lipid. So it has streptavidin, a gold particle. And at that point, when you shine the laser beam on the streptavidin, it absorbs the energy and it releases it locally as heat. So in this case here, when you see the movie being played, you'll see the two membranes come together. They form a VIB, a stone membrane. And now if you shine the laser pulse at the point of contact, the two membranes fuse and you bring them back. Can you still hear me okay? Oh, sorry, hit the wrong button then. And then you can then start to make kind of tissue-based systems where you systematically fire the laser pulse at each of the different interfaces. And that means you make vesicle A fuse with vesicle B, then AB fuse with C, ABC fuse with D, and you can control the sequence in which things come together. Last couple of slides. I showed you the kind of compartmentalized systems before. These ones are called multisomes. So these are basically what I call the fatty versions of the vesicle. So imagine, as you form a two-compartment system, if you were to pump the void between the two monolayers with oil, you would get this version here, which we call a multisome. And again, these are stable for weeks. And on chip, you can make these up with up to 20 different subcompartments. And we've shown again, we can get controlled kind of reactions between these here. It's like just a pyridium with a primary amine to form pyridium, but you can do much more complex reactions in these subsystems. You saw some really nice work in the previous talk about the use of light. So we use that as well to kind of trigger these enzyme cascades. So here's an example of a nested system where you've got small vesicles inside a larger GUV. And in this case here, the lipids that make up the smaller nested vesicles are made up of a lipid called DC89PC, which when you shine light on it, it polymerizes and becomes porous, whereas the outer membrane is unaffected. So this is a way of actually separating, rather than kind of using channel proteins, separating enzymes from reactants or vice versa, or enzymes from enzymes, by basically, when in the absence of light, they can't see each other. When you shine the light, the lumen then comes into contact with the subcompartments and you trigger enzyme cascade. And again, that's proven to be a really controllable kind of thing. So the last part of my talk, I just wanted to talk to some more recent work in the last few years where we're moving towards what we tend to call cellular bionics, where we do a lot of work, which I've not got time to talk about, unfortunately, about getting real cells to talk to synthetic cells. But the example I'll give you now is more about an ensemble where synthetic cell and real cell work together in kind of tandem. So a couple of slides ago, I showed you an enzyme reactor that had 3 subcompartments where you had 3 different enzymes in each subcompartment. It will take in lactase and produce rhizorufen. And we thought, hang on, could you make a system where you take a synthetic cell and you put a real cell inside it? So in this case, it was a cancer cell, a BE cell. And again, we do this on chips. So every synthetic cell that comes off the production line has, some have got 2, but in most cases, one real cell inside it. And with that cancer, because we work with them a lot, you can control the protein expression. So the first part of the enzyme cascade, which is beta-glucosidase, we make 2 versions of the cancer cell. One version of the cancer cell that expresses it, and one version that doesn't. And then you load up the synthetic cells with these cancer cells. One population can express the first part of the enzyme cascade. The second population can't. And then what happens is when you then provide the feedstock for this kind of ensemble, as long as the cancer cell is alive and viable, it takes in the lactase, and it basically triggers the cascade and you produce rhizorufen. So that works in an ensemble. And then when the cancer cell dies, the ensemble stops functioning. And in the other version, where you've knocked out beta-gal, when you provide the lactose, nothing happens. And we've done some more stuff recently where we've actually started to do transplants where rather than put whole cells inside organelles, we take whole cells inside synthetic cells, we actually extract organelles from real cells and put those inside the synthetic cells and kind of get combinations working like that. So this is kind of just the data kind of showing how the ensemble works. And when you basically have beta-gal knocked out, the fusion between the real and living cell, so the synthetic cell, the real cell doesn't take place, but when it's present, it does. And with that, I just want to summarize that there's been a lot of work last few years, particularly in the area kind of biomembrane engineering, that allows us now to make the kind of chassis that we need to make the synthetic cells. I think a lot of the work that's now happening is based upon really fundamental understanding that's been in place for a long time, but it's now being used to kind of stitch together circuits that are based upon really quite well established knowledge, but that hasn't been applied to the synthetic world. And I think the last piece that for me is very exciting is the technology that you need to do this really is not obtrusively expensive anymore. I think the world of microfluidics was in the domain of people who had access to multi-million-pound clean rooms, but you can now be, an expert in microfluidics for a couple of $100, you are really in the game. And I really encourage young researchers to move in that direction. And finally, I'd like to thank all the following people for their help, in particular, Yuval, Nick Brooks, Rob Law here at Imperial, Paula Booth, who's at King's College London. And finally, thanks to all my funders and you for listening. It's been an absolute pleasure. Thanks, folks.
Thank you, Oscar and Hannah. Terrific talks, and we're going to open it up for questions. Everybody, please send your questions in. And while they're arriving, Oscar, I have a question for you. So much of mechanosensitive coupling in biological membranes involves spectrin membrane skeleton. But you're working with small vesicles. Is it possible to reconstitute sort of a mini membrane skeleton inside of these vesicles, possibly through anchoring, so that you could actually study the role of the membrane skeleton in the mechanosensitive coupling?
It's a really good question. I've been asked that before. So we've not tried to do that yet, but I do know there are groups who kind of have come from that side of the spectrum and are now applying vesicle technology to kind of reconstitute that. So it's something that people are currently trying to do. It's not something we're doing, but it's definitely something that I know a number of groups are on the case with. I think once that's up and running, it'll be really nice to see how the two kind of couple together.
Thank you. So have we received questions from the audience, Jess?
I don't have any questions either in the Zoom or the Facebook Live. We can give everyone a minute or two if you have any pressing questions.
Maybe
Otherwise,
I
of
can
course.
ask a question. Oscar, that's so fascinating. So I was wondering what kind of organelles you've transplanted into these synthetic cells. Like
Things like
mitochondria, Golgi. What is it that you
so at the moment we focus on things like chloroplasts. So as energy sources, because they're kind of, when you take them out, they are robust for, again, it's stuff that it's not yet published, so but I don't mind talking about it. But it's, as a chemist, I was surprised how long lived a chloroplast can be when you take it out of a cell. 12 hours, 13 hours quite easily. So that provides basically almost like a bio battery for a synthetic cell. So at the moment, we're just trying to see how far you can push that approach in terms of taking kind of organelles and putting those together. And then, like I said, the other part of this that's kind of similar to that is the work around synthetic cells and real cells talking to each other. But I didn't have a chance to work today.
I see questions coming in. Jess, you want to ask the question?
Sure. We have one from Dr. Thompson. How do bar domain proteins impact the interaction of the vesicles?
Was that one for me?
You didn't specify.
Anyway, so I showed at the start of my talk different kind of proteins that can be used to. I think I gave the example of an M-bar as an example of a protein that we know interacts with membranes and kind of regulates curvature. We ourselves haven't put these in synthetic cells yet, but I mean it is an example of something that could be done, but it's not a motif that we've explored yet. But I think it'll be from particularly when we work with small nested vesicles, it would be really cool to try with that. Those are kind of the length scales of kind of 50 to 100 nanometers, a way of kind of controlling their kind of size. But we've not done that yet. But it was just an example of, I think in the literature there must be 20, 30,000 papers of membrane X does this to protein Y, and the N-bar domain is probably one of the most studied, but it's not one we've gone there yet.
Jess, do you have additional questions?
That's all that's come in so far. And of course, we can pass along any questions you may think of post-event to the speakers. Today, you're welcome to email us at ed@scbm.org. Oh, yeah.
I went to Hannah actually. So Hannah, the de novo protein that you showed that had the lid. What's the biggest type size of molecule you can actually? Does that actually have a channel that you can? How big is the object you can actually pass through that channel?
So it's actually not so I guess you're asking about the kind of the peptide that you can thread on this.
Yeah.
We haven't really pushed that very far yet. I mean that degron is pretty small. It's a CODC degron, so it's relatively small.
Yep.
And every time, so you just you just thread it on the latch. You're not stuffing anything inside that molecule really. And also, you know, every time you want to thread a different peptide in there, you have to redesign the latch, tiny little
Okay.
latch. But it could be done computationally.
But in principle, you could make something that has a larger kind of lid that actually is
Yeah,
almost like chemically non-specific if you wanted to release a wider cargo. That's
Mhm.
really fascinating stuff.
Mhm.
Pretty cool.
Yeah, yeah, yeah.
So I actually have a question for Hannah also. This one's a really crazy one, Hannah. But in terms of what you created, how is it impacted or is it impacted by the ubiquitin proteasome system within cells?
Yeah. So, so the data I showed you was in yeast, and so that's, you know, this isn't a problem, but it is possible. I mean, one thing that we kind of thought about and actually did quite a bit of experiments to figure out is whether the basically this overexpression of this of the switch and the fact that it needs to be degraded and so on clogged the actual proteasome system there, and it didn't. You know, we went up quite a bit in concentration and it still didn't. Now, of course, we put those constructs in mammalian cells and various mammalian cells, and there also we really didn't see. I mean, we didn't measure what the exact crosstalk with the ubiquitin system, but we really didn't see, at least in hex cells, for example, a huge impact at all on cell viability or anything like that. So
Thank you. Thank you,
that
Hannah.
doesn't answer 100% your question, but yeah.
I appreciate it. So any further questions, Jess?
Nope,
Well,
none more in the chat boxes.
then I want to thank both Oscar and Hannah. Those were brilliant talks. Very much appreciated. And thank you, the audience, for being with us today. So long, everybody.
Real
Thank
pleasure.
you all.
Take care. Nice people. Take care. Bye.